Welcome to our lesson on the understanding of genetics. The key questions for today are: what experiments did Gregor Mendel carry out? How did Gregor Mendel advance our understanding of genetics? What work after the death of Mendel validated what he had found out? What is our current understanding about genetics and genes? How does Mendel's work relate to our current understanding? The key terms that you will need to know are dominant, recessive, gene, chromosome, validate. In the mid-19th century, Gregor Mendel was a monk and carried out breeding experiments with pea plants in the Abbey gardens. He kept meticulous records of his techniques and observations, which, after his death, led to him being recognized as the founder of genetic science. Mendel crossbred pea plants of different colours and observed patterns in inheritance. He theorised that characteristics were passed on through units from the parents to the offspring. He realised that some of the units could be present but not expressed. He found this in breeding white and purple pea flowers. The first generation showed only purple flowers, but when those were bred, the next generation showed 75% were purple, but 25% were white, showing that the unit that was responsible for the white colour was still present in the purple plants, but not expressed. Task: how could we explain these observations using what we know about genetics now? We now know that genes are responsible for characteristics, and they come in two forms, dominant and recessive, which interact in the way that Mendel observed. In this example, we would see the genotypes of the parent plants as capital P for the purple and lowercase p for the white. We know that the purple parent is homozygous for purple, as all of the first generation offspring were purple. This can be shown in a Punnett square. All of the offspring have the genotype capital letter P, lowercase letter p, meaning they are purple as the purple gene is dominant. When this generation are bred together, the offspring show the presence of the recessive white gene. The offspring will have the genotypes capital P, lowercase p, and capital P that would be purple, and the genotype lowercase p that would be white. They are in the ratio of purple to white of 3:1, so 75% purple and 25% white. This was consistent with Mendel's observations. Later in the 19th century and in the early 20th century, chromosomes were observed during cell division, which validated Mendel's theories and led to the understanding of what we now know as genes. In the mid-20th century, further technological and scientific advancement allowed the structure of DNA to be determined and the mechanism of gene function worked out. Task: why was the importance of Mendel's work not recognised until much later? Mendel was a relatively unknown scientist, and there was no evidence for his claims. It was not until later, when DNA, chromosomes and genes were better discovered, that Mendel's work was understood and therefore recognised as important. Extension: answer this AQA GCSE exam question. The diagrams show one of the experiments performed by a scientist called Mendel. He bred sweet pea plants. In the sentences below, cross out the two lines which are wrong in each box. Mendel proposed that flower colour was controlled by inherited factors. The first generation plants show that the red factor is. The second generation plants show that the white factor is. We now call inherited factors. These factors are passed from generation to generation in. The red-flowered sweet pea plants did not all grow to the same height. This was due to factors. Mendel proposed that flower colour was controlled by inherited factors. The first generation shows that the red factor is dominant. The second generation shows that the white factor is recessive. We now call inherited factors genes. These factors are passed from generation to generation in gametes. The red-flowered sweet pea plants did not all grow to the same height. This was due to environmental factors. Task: Mendel's crosses of red flowers produced 5,496 red flowers and 1,832 white flowers. Explain why Mendel's crosses gave him these results. Red flowers must have a genotype of capital R, lowercase r, so they were heterozygous. This means 75% of the offspring would be red and 25% white. There were 7,328 offspring in total. 5,496 is 75% of this, and 1,832 is 25% of this. In a Punnett square, this would show as 3:1 ratio.